सबसे पहले प्लांट डीएनए एक्सपेरिमेंट्स, ब्लड डीएनए एक्सपेरिमेंट, सलाइवा डीएनए एक्सपेरिमेंट, वट एवर डीएनए एक्सपेरिमेंट, देर आर टोटल 7 steps, right? First is sample selection, second sample homogenization, sample cell lysis, then protein precipitation, then DNA precipitation, then DNA desalting and washing, and then DNA storage. आपने पूरी प्रोसेस कंप्लीट किया. अब आइए सैम्पल सिलेक्शन में. सॉफ्ट एंड न्यू, इजी टू हैंडल, इजी टू ग्राइंड, इजी टू सेल लाइसिस, फ्री फ्रॉम कंटामिनेशन, राइट? Then come to the cell homogenization. That means you have to break the cell uniformly. That's homogenization process. Temperature, sound, stroke you will keep low, as best low possible, right? Then come to the cell lysis. It's like masala fry. Every scientist here change the chemical composition, little procedure, sample to sample, right? जैसे जितने भी सब्जी खाना बनाने वाले लोग होते हैं, मसाले उतने ही होते हैं, लेकिन उनका कंपोजिशन घटाते बढ़ाते रहते हैं. प्रोसेस थोड़ा चेंज करके नया टेस्ट डेवलप करते हैं. वैसे ही यहाँ मॉलिकुलर बायोलॉजिस्ट क्या करता है, सेल लाइसिस का सोल्यूशन बदलता रहता है, क्योंकि सेल बदलती रहती है तो उसका कंपोजिशन भी बदलना पड़ता है, राइट? तो यहाँ पे बदलाव आपको प्रोटोकॉल्स में मिलेगा, तो परेशान नहीं होना. थोड़ा मोड़ा ऊपर नीचे सोल्यूशन की कंसंट्रेशन अप डाउन जाएगी. आफ्टर दिस देर आर टोटल 4 steps. All is common for plant, blood, hair, saliva, same. Protein precipitation same, DNA precipitation same, DNA washing same, DNA storage same, right? Then protein precipitation में क्या यूज़ करेंगे? Chloroform isoamyl alcohol that precipitate the protein by mixing, inverting the tube. Then you come to the next part, DNA precipitation. As soon as you add DNA, it get precipitate thread-like structure is come out. By tip, by naked eyes, by putting it this way or turning this way, you can see threads of DNA is visible clearly. When you do the centrifuge after that, DNA stick at the bottom, liquid will remain up, tube open, discard the supernatant, save the pellet in the bottom, means DNA. Let it dry completely. Overnight, put open tube, let dry. Once it is dry, add DNA storage buffer. I added from that tank. Tank have same solution, trace EDTA. That is DNA storage buffer solution, right? In that DNA dissolve. Again, a small quantity of dye mix, that's called DNA loading dye, made up of bromophenol blue cyanol FF, written in your literature itself. If you see DNA loading dye, you will find out the composition. Small 2 microliter dye, 10 microliter DNA mixed together and load into the electrophoresis. Once you load it in electrophoresis, DNA will run in electric current. You see there we put the electric current. That current makes the traveling of DNA according to their size and separate it according to size. As you see in the DNA fingerprinting, many bands is coming. When you do the DNA fingerprinting, multiple fragments may be generated by different size. They can separate out according to their size. So you can find out what DNA, what gene, what kind of their size, what kind of their role, right? By profile study, you can identify characters, genes. By matching the DNA, you can find out their ancestors. Relationship can be predicted easily. Understand? This way, total DNA, molecular biology, biotechnology, 90% chapter is over. After that, you start DNA fingerprinting, cloning, and genetic engineering work. What you do if you want to genetic engineering, you just use 2 enzymes. One is restriction enzyme that cut the DNA. One is joining enzyme that join 2 fragments of DNA called ligase. 2 enzymes you will use. DNA cutting and joining procedure start every day in your lab. Cut and join. You will be expert in DNA, genetics, engineering. Then further transformation. When you cut and join DNA, where you put inside which body? If you transfer that DNA in that body, means you did the transformation. Means your genetic experiment successful, right? And then further you can go for DNA fingerprinting. In that DNA fingerprinting, you will do PCR experiment, real-time PCR experiment, that in COVID time. अब now you can be a scientist, a different subject specialist. If you are forensic scientist, if you are breeder, if you are molecular biologist, if you are identifying cancer, if you are identifying any mutation, so your role will be different, but the image will be same. Procedure will be same. According to your objectives, you find out your result from that image. If you know the image analysis of DNA, you are perfect molecular biologist, perfect DNA scientist, perfect in biotechnology. That will be enough for your career point of view. Second important step, listen, student, whatever you doing, whatever subject you doing, whatever course you doing, whatever you want further, try to make the product. Products will give you the job. Company क्या करती है? What company do? Company gives the jobs. How they gives the job? They makes the product. And if they not make the products, they do the transportation, they do the labeling, they do the courier, they do the loading, unloading, they do the printing their boxes, like that, okay? To make the products, process, sell out, consume out, so multiple jobs generated. If there is no products, no city, no life, no status, nothing. If you have products, you can make Gurgaon type city. You can make Greater Noida type city. If you don't have product, you will be a small district, come, reach to the roadways, jump out of the city. Understand? In my city, in your city, throughout the India, if any big city you know, there is product maker. And a small city, there is nothing like Mathura. If you come close, see multiple gold business is there. They generating huge amount of money. In Delhi, there is a 1 kilometer area. It's called Mayapuri Industrial Area. Generate 6,000 crore rupees. 6,000 crore rupees equal to 1200 MP MLAs of the country. कितना? 1200. उत्तर प्रदेश में कितने हैं? 403 MLA. राजस्थान में कितने हैं?